Hello everyone, welcome to my Study Tech YouTube channel. In my previous video, I have demonstrated how to make this VCF file from the BAM file. Today, I will demonstrate how to install the snpEff tool and make the result from the VCF file. You can follow this pipeline for genomic variant analysis. Check out my previous all video. So in this video, first I will demonstrate how to install the snpEff tool, and second, how to download the annotated database of Saccharomyces cerevisiae and how to annotate the VCF file and make the result. So let's start with installation of snpEff tool. So go to the Google and write snpEff and enter. So go to this site. So here is the download button. Let's download it. It is about 63 MB of size. So download is complete. Let's check it. So first you have to extract it. Okay, extraction is done. So to run it, you can see here is the snpEff.jar file. So simply open a terminal here and let's check it. So you need Java pre-install to run this snpEff. You can check out my Java installation video to install Java. So to run it, Java, then jar, then you have to write this command and enter. So now you can see the whole usage of this tool. You can just copy it and make note. Okay, so here is the VCF file of my previous analysis. Now we need the database. So here is the Saccharomyces cerevisiae VCF file. So we need to download the annotated genome. So for that, just clear it. You have to run Java, then jar, then snp, and after that you have to write data bases. So it will show the database. Now I want to just download all the file as a database.txt. Let's enter. So let's check it. So here you can see the database file is generated. So let's check it. So it is a long list of database. Let it open first. Okay, so my organism is Saccharomyces cerevisiae. So I will try to find it. Control F and let's enter. It may take time. Okay, See, here is the Saccharomyces cerevisiae file. It is the actual name of this Saccharomyces cerevisiae database. There is only underscore. So let's copy it and let's write down into your note. So you have to download this database to run your VCF file. So to run it, go to the terminal, just clear it again, and again java jar snpEff, and then you have to write download, and you have to paste your organism entry name. It is Saccharomyces underscore cerevisiae. So your organism should be in this database. Let's download it and enter. Okay, download is complete. Let's check it. So here is the new directory called data. You can check it. So here is Saccharomyces cerevisiae directory. Here is the database file. So I have the database file. So I can now run this VCF file. Let's cut it and keep it in this directory to run it. So let's check it. So here is the VCF file, and we also have the data of cerevisiae. So to run this VCF file into Java, then you have to add one Java parameter, that is x, capital X M 8G, then again jar, after that snpEff.jar, then v, then canon, and after that you have to type your database name, Saccharomyces cerevisiae. Let's paste it here, and in result I need the csvStat. So for that you have to write this stat. So the stat name is result, and now I have to write the file name. So it is the file, input file, and I am to convert to it almost equal name. Here I am adding annotated VCF file. So it is the total script. Let's enter. Okay, I just did a mistake here. So there is a command is x here. Okay, so it is the command. Let's enter. So now it is working. Okay, done. Let's check it in terminal. So now you can see here is a much bigger VCF annotated file. Okay, let's check it in directory. So here is the annotated file, and this is the result file. So it is a txt format of result file, and you can also see the HTML version of result. Just open it with your browser. So here you can see this is the annotated database, cerevisiae, and the chromosome wise variant rate, how many SNP is there. This is indel, insertion, deletion, some mixed type of. This is the all count. You can use this result file as your result. Here is the other parameters, you can check it. So, and here is the annotated file. If you found this tutorial helpful, please give it a like and leave a comment. For more bioinformatics tutorial, don't forget to subscribe to my channel. Thank you.